Good evening, everyone. Next, xenotransplantation. Xenotransplantation. What is xenotransplantation? Before coming into that, why it is in news? If you have heard that there was a case in which the pig's heart was transplanted into a patient, and after surviving for a few days, that patient died. So the thing is that normally we have heard regarding so many surgery, we have heard regarding organ transplantations, right? That if you have some kind of failure in your kidney, we will search for donors. Yes, if everything is matching, then we will get a kidney. We have heard about heart transplantations. We have heard about stem cells that are giving rise to some kind of heart and all. But xenotransplantation is something in which it will transplant or it will implant an animal's organ into a human body. Okay, it is very, very important. So in simple terms, have you understood what is xenotransplantation? Right? And for these particular, for the xenotransplantation process, mostly pigs are being used, genetically modified pigs. Okay, so much GMO pigs, genetically modified organisms here, they are the genetically modified pigs, right? Now the case is that this modified, why pigs are being genetically modified? Any idea? Can you share with me if anything is coming into your mind, why the pigs are being genetically modified? Basically, see, the pigs are being modified so that these pigs will produce some kind of human proteins. These pigs have been modified in such a way that these pigs start producing some kind of human proteins. If human proteins are being made by these pigs, then if that organ is being transplanted, if the heart of the pig is transplanted, then there will be less chance that our body will reject. Yes, normally the most important problem that is associated with organ transplantation is graft rejection. Do you remember? Do you remember about graft rejection? Yes, even if it's in case of human, we have found this problem, isn't it? Graft rejection is a major problem. Now, in order to subside this graft rejection between you, when we are taking some human organ, what do we do? What is done for removing or for reducing the graft rejection? Do you remember immunotherapy? In immunotherapy, we have two different types. One is activational and another is suppressional, right? So that means here what we will have to do, we will have to suppress the patient's immunity so that there is less chance of rejection. We cannot activate it. If we activate it, it will reject it more. Where do we use activational immunotherapy? In cancer, right? In cancer, we use activational immunotherapy in these kinds of diseases where some unwanted masses, unwanted cells or unwanted things are present. But if we have to suppress our immunotherapy, then we will utilize it in case of this organ transplantation. And second is the allergy, right? In allergy is somewhere in which some other agents, some non-harmful agents will be accepted by the body as harmful manner, as harmful things. So during that part, we do not want the reaction to happen. So we will suppress the immune system, right? Anyway, coming back. So if in between humans, if there is rejection of organ, then definitely there will be rejection of organ. The graft rejection will also be present in xenotransplantation. Am I right? This is the main problem that is associated. So of course, suppressional immunotherapy has to be adopted. But apart from that, some kind of modification is also done so that that particular pig is producing the protein similar to that kind of human. Now, another thing, we have seen these advantages, right? But the major disadvantage, if you see, if the pig is infected with retrovirus? Yes, that retrovirus, as it comes to human, there could be pop up. Maybe, suppose, in case of the pig, a particular virus was hidden. It was present, but it was not showing its color. It was in its latent condition or its hidden condition. Now, as it has moved to the human body, human never know, maybe human body might give some kind of favorable conditions. If human body is a favorable condition for the virus, then maybe there could be spread or there could be disease related to that particular virus. So this is another very, very important or major disadvantage of xenotransplantation. Now the question is, we have so many organisms, right? Genetically modified organisms. We have cows, we have monkeys, apes, and we all know these chimpanzees are more closely related to us. But in spite of the fact, what we are using pigs. Why we are using pigs? Why only pigs? This is because this pigs, the size, the organ, when you are modifying the pig, okay, the organ that will grow bigger, it takes very less amount of time for a pig's organ to grow up to a size that is required by a human. Remember this point. There are several reasons, but one very important reason is that the pig's organ to grow to a size up to a human organ. This is very much feasible in case of pigs. They require very less amount of time. Point number one. Point number two is that I think you all know pigs, when they give rise to babies, lots and lots of litters are being produced, right? This lots and lots of progenies or offsprings, lots and lots of pigs, small baby pigs will be formed. Am I right? I think you all know this thing. But this thing is not seen in case of chimpanzees. This thing is not visible in case of monkeys, because they are also like us. They are also like humans. They will not give rise to huge amount of population at one time, isn't it? So the thing is that the pigs, they produces a lot amount of offspring, and therefore when they are producing lots and lots of offspring, the testing, this genetically modification, whether it's taking place or not, this checking, everything becomes feasible, and therefore pigs have been chosen as the best candidate for xenotransplantation. Vincov-19. There are so many drugs available for COVID-19, but this Vincov-19 is in news because it's the first indigenous COVID-19 drug that will be made available soon. Okay, the first indigenous COVID-19 drug. So you can see here, molecular biology, University of Hyderabad, the department. So they have collaborated. These are not that important, but anyway, talking about Vincov-19, what we need to remember is that here, how this Vincov-19 has been made. That is something that we need to know, right? The first step is that this virus has been inactivated. If you remember, we need to, in case of vaccines, of course, this is not vaccine, these are drugs. In case of vaccines, when we were using the organism, either we were using the live organism or we were using the dead or killed organism. When we were using live organisms in case of vaccines, we made it weakened. Do you remember? They were being attenuated, weakened or attenuated. Am I right? Yes. And in case of dead or killed organisms, dead virus, we made them, not only virus, it can be a bacteria as well, either heat killed, chemically killed, or it's killed by any kind of radiation. Similarly, if you have to develop a drug as well, if you want to develop a COVID-19 drug, definitely you will have to take either a component of the virus, you might take a subunit of the virus, or if you are not taking the subunit of the virus, then what you will have to do, if you are taking the entire organism, if you are taking the entire virus, you will make it inactivated. After making it inactivated, how to make it inactivated? With the help of heat, chemical, or any other radiations. So then you will inject it into horses. As the same thing like the vaccines, as they are injected into the horses will recognize that this particular thing that is coming into my body is not my own. Yes, are you getting my point? Just like vaccines, new foreign things are coming. If new foreign things are coming, what will happen? The horse body, it will start preparing the Indian army. And what is that Indian army? The Indian army is nothing but the antibodies that will be developed everywhere. Am I right? And these antibodies that are generated, these antibodies are extracted. Okay, the antibodies that has been produced by the horse in response to the inactivated SARS-CoV-2 virus that will be extracted, and then it will be purified, and these purified antibodies have been developed, are being marketed as Vincov-19. Similar to kind of vaccines, but here, since we are extracting out the antibody, right? And that too not of human, that of horse. So we are calling it as drug. And by drug, we mean drug, it could be a chemical, right? Antibodies are chemical antibodies. Basically, they are biochemical antibodies. So it's better if we call them as generic drug. Now, if you see, have you heard about plasma therapy? Basically, if I have to tell in simple language, plasma is a part of our blood, right? Plasma. It's a part of the blood. Blood is liquid. It comprises of blood cell. And this plasma is the liquid portion of the blood without the blood cells. Blood cells means red blood cell, white blood cell, and the platelet. So without this blood cell, if you take out the liquid portion of the blood, that is the plasma, and the color of the plasma, trust me, so is it red or not? Can you tell me whether the color of the plasma will be red or not? It's not red, right? It's not red. Why it's not red? Because we have removed the RBC, isn't it? We have already removed the RBC, and the RBC contains the hemoglobin, which is red in color, red blood cells. Am I right? Hemoglobin, it's red in color because of the presence of a iron pigment, heme. Anyway, so as we are removing the cells, the plasma will not be red, it will be light yellow in color. That means if you take a test tube, okay, if you take a test tube and if you give blood here the cells of the blood will be present. Okay, the red blood cells will be present. Next, you will find some white blood cells, and finally you will find a kind of yellowish component, and this is the plasma. Am I clear? If you separate blood into its component in a test tube, this kind of picture you can visualize. Now, as we are talking about plasma therapy, isn't it? As we are talking about plasma therapy, if you remember, there were lots of situations in which people were asking for blood. People were asking, suppose even there was a time when I also suffered from COVID-19. People were asking me if I could donate blood for plasma therapy as well. Why? Why plasma therapy? Because as I was infected with COVID-19, That means my body has produced antibody in response to the virus, right? Antibodies have been generated. Now they will take my blood, they will purify it, they will take out the plasma, and in the plasma there will be the antibody, and that plasma containing antibody will be injected to another patient who is suffering from COVID-19. As a result, if it's injected to a patient, to another patient who is suffering from COVID-19, what will happen? More and more antibodies, it will act like a medicine. Am I right? These antibodies from me, if I'm giving my antibody to other person, will it act like a medicine? Yes, the antibodies will fight against the virus. So similar kind of situation we have seen in case of Vincov-19 as well. The only difference is that here we have injected the virus into the horse. The antibodies have been purified. It have been extracted from the horse. So it's called as, so it's a kind of sophisticated scientific plasma therapy.